for the opportunity to present our work here. My name is Anand Ramasubramanian. I'm an associate professor of biomedical engineering.
Say that again. I want to hear you say that name.
So it's fairly easy, actually. You break it into three pieces: Ramasubramanian. Yeah, it looks ominous, but it is not as bad as it sounds. That's right. So this work was predominantly done by one graduate student, and I'll talk about what he has done and with collaborators. I'm an engineer, so we have really good collaborations with microbiologists, and I still can't differentiate between gram positive and gram negative. I get confused. So this is a microbial threats. This is a report from CDC in 2013, and you may have heard of the escape pathogens. And particularly, I will be talking to you about Candida, and particularly about the fluconazole-resistant kind, the bifilms. And then I will talk to you about Pseudomonas aeruginosa and also Staph aureus. And what I'm going to talk to you about is both new technologies for drug discovery and also for diagnosis. So the drug discovery, obviously we do need new drugs, and apart from rational use of antibiotics, but also we need really effective antibiotics too, which are preferably targeted. In general, the drug discovery pipeline goes like this. So starting from target identification, the targets are validated, and then screen libraries to get hits, and from hits you zero down to few leads. The leads are optimized by chemical optimization. They're tested in preclinical models. It could be in vitro cell culture or animal models, and then they go on to clinical trials as phase 1, 2, and 3. So this is a long and arduous process, and in general, the drug discovery process can be classified roughly as either rational or irrational. The rational is when you have a 3D structure of the target, and then you do computational modeling to obtain leads that exactly fit the target and then stop the action of the target. It could be enzyme inhibitors or whatnot. The other case is irrational, or what we call basically is a high throughput screen, where you don't really need a structural information, but instead you screen really large libraries of compounds against, let's say, a pathogen of interest. So you don't really care about what the targets are, what the lead is, and then you just screen it out, okay? And then this obviously means that this is going to be like searching for a needle in a haystack, all right? So in the for the high throughput screen, typically the first high throughput screen was done by Pfizer, and it took them about 1.5 years to screen 100,000 samples. This was way back in 1952, all right? And since then, from flasks, we have moved on to well plates, such as 96 well plate or 384 well plate or 1536, and now we can screen more than 1,000 molecules per week. This is a really low estimate. So, for example, with our collaborators, we did some screening for against Candida. We grow the Candida cultures overnight, and they are resuspended in media, and they are transferred to a 96 well plate containing compounds. And then you let them grow, and then you add a viability dye, and based on the dye, you essentially decide whether you have cell growth or not, okay? So it looks more or less like this. So if you have no drugs, that a lot of things grow. If you have drugs, you have an inhibition. And by looking at the fluorescence or absorbance in this case, you can know whether a drug is effective or not. Obviously, this is a really long process. So the alternative that are coming up these days with a lot of development technologies, such as lab-on-a-chip technology, are microfluidics and microarray-based assays. So today I'm going to talk to you about the microarray-based assay that we have developed. So I'll talk to you about the design of the microarray and how do we do the readout, and then what are the advantages of using a microarray-based high-throughput screen are. So we call it CAB chip because it is for Candida albicans biofilm chip, and everything is done on a standard microscope slide, basically 1 by 3 inches. Okay, so this is how essentially it looks. You have all these different spots, and then on a single microscope slide. So what we do is we take the yeast, cells, and you mix it with a hydrogel. I'll talk about this in a second. It could be collagen or alginate. That's what we have used. We mix it with yeast with the hydrogel, and the media that's necessary for the yeast to grow. And using this robotic arrayer, we spot the cells at different spots on a glass slide. And then the glass slides are incubated in high humidified conditions for 24 hours to allow for the biofilm to form. So as you can see, it essentially, so you can see all these little dots, and each of them is basically a culture in itself, all right? And after 24 hours of culture, we take it out, put it back on the robotic arrayer, and we spot the drugs on top. We let it incubate for 24 hours and for the drug to act. And after it is done, all we do is we take the entire slide and dunk it in a Falcon tube containing buffer to wash off the excess drugs. And then we stain it with a viability stain and stick it to a DNA microarray scanner, and you can read the output. Okay, so there is no media addition in terms of pipetting. Nothing is needed. So everything is done semi-automated. All right. So what are the specifications for the cap chip? So so as I said, when it forms these cap chips, you notice that it sticks onto the glass slide because you want it not to fall off when you're washing it or during processing. And importantly, they have to be true biofilms. It cannot be planktonic because these are encapsulated in gels. So we have to make sure that they form true biofilms with all the morphological and the antibiotic resistance characteristics of a biofilm. And we do these assays for about 2 days. So you do not want the cells to dry up, and you want the cells to be alive. Okay. So to cut the long story short, what we modified the glass slides using chemical methods. We have published this, so I will not go into the details. But the idea is to essentially chemically link the matrix to the slide and allow the biofilm to form within the matrix. So that is basically the idea. Okay. So this is how it will look. This is the glass. So you have the glass slide here, and on top of it, you have a nice hemispherical spot containing the cells which grow into biofilms. Okay. So this is the side view of the spot, and you can see that it is really hemispherical. And when you look at the SEM pictures of this, you can see that the candida has indeed formed these morphologically, the hyphae, which is essentially the biofilms. Okay. So with this system, we were able to spot 1,200 spots on a single slide, which means you have 1,200 different cultures, and each spot has a volume of about 30 nanoliters, and the starting density is only about 90 cells, and if you can go lower if you want and allow it to form biofilms. And once we stain it, we can do a scanning with a microarray. So the stain is, if the cells are live, it is going to stain green. If the cells are not alive, it will be less green. Okay. So here the cells are all alive, and this is how each spot looks. And when you go closer, you can see that these really nice hyphae characteristic of a Candida albicans biofilm. And when you look at it sideways in a three-dimensional pattern, you can see that it does indeed form these three-dimensional structures. Okay, and this is a confocal microscope of the. We have stained here not only the hyphae but also the exopolymeric matrix, and together you can see that it has formed these really nice thick biofilms in each of those single spots. So this is how it looks over a period of time. This is the growth kinetics over 48 hours. We start off with single cells, and then over a period of 4, 8, or 12, or 24 hours, by about 12 to 24 hours, it forms these really nice biofilms. And then at about 16 hours or so, we add the drugs. About 24, we could have added earlier, but we wanted to keep it 24 just to be consistent with what has been done in the literature. So 24 hours later, we add the drugs, and we essentially let it grow for another 24 hours and see the action of the drugs. Okay, so these are just a couple of examples. So here we did two types of experiments. One is preventing biofilm formation, or is it for preformed biofilms? So preventing biofilm formation, what you do is you mix the drugs with the cells and spot, And in the case of preformed biofilms, you allow the cells to form the biofilm and then you expose them to drugs. Okay? So I'll tell you why this is important. This distinction is important in a second. So amphotericin is a very commonly used antifungal, and you can see that from our system you do have a nice IC50, which is comparable to a standard 96-well plate model used in clinical laboratories. And again, the same thing with caspofungin, which is a very potent antifungal. And in the case of fluconazole, you see that the preventing biofilms, fluconazole is effective in preventing biofilm formation. That is, if the biofilms are not formed, the cells die when you add fluconazole. But if the biofilm is already formed, the fluconazole is not effective, which is another characteristic of the biofilms. The biofilms are resistant to fluconazole. That is well established. So our system, these data again shows that what we have is a very viable biofilm culture platform which one can use for drug discovery or for diagnostics. So I will skip this part. So I have 10 minutes gone so far? Okay. I'm okay. Is it 10 minutes now? Yeah, about 10 minutes. All right. So I guess I will discuss this data that we have here. So as I said, fluconazole resistant Candida albicans is a serious threat. However, it turns out that fluconazole can be combined with certain drugs, and that would make it much more effective. And fluconazole is a very preferred drug because of low toxicity too. So in this work, what we show here is we use the screen, we use the cap chip screen to essentially look for combinatorial screening of FK506. FK506 is tacrolimus. It is an immunosuppressant usually used in the clinics. And we looked at a bunch of antifungals from this library called Prestwick Library. Prestwick Library has a whole bunch of FDA-approved compounds, including antifungals. They may not be used in clinics, but they are all FDA-approved. So we want to see if FK506, which is usually given to immunosuppressed patients, combining FK506 with any antifungals will improve the activity of the antifungals. Okay, so here we have FK506 at different concentrations, starting from 0 to high, and along the different rows we have different antifungals. Okay, so you see that here the percentage of inhibition. You see that when there are certain drugs which are effective without even adding any FK506. Okay, however, when you add FK506 at really low concentration, let's say number 4 microgram per mil, you have certain other antifungals that become effective with FK506, an example being fluconazole. And this has been shown by previous work that FK506 and fluconazole are together effective. So in this case, we have shown that it is again benchmarking our assay that you can combine fluconazole with FK506 to have effective anti-combinatorial therapy. And so we did identify a number of different compounds here listed in blue, which may be helpful as a combinatorial drug for antifungal therapy. And we went on to screen another library. This is from National Cancer Institute. It's a challenge set. It had about close to 100 compounds, and we used all the 100 compounds on a single chip, and we were able to screen and compare it against 96-well plate. Okay, so in essence, what I've shown you here is that we were able to develop a platform, a microarray printed platform called a cap chip. And compared to a standard 96-well plate, it is certainly a lot more advantageous in terms of the volume or the speed at which you can do, and the ease with which you can do this work, and also the sensitivity because of the fluorescence with the microarray scanner, and it will be cost effective too. So moving on, so that was what I showed you is about Candida albicans. So we moved on after finishing this work to bacterial or pathogens too. So first one here I show you real quick is a Staph aureus chip, and here we spotted in a very similar fashion. We did certain optimization and we spotted Staph aureus, and on the same chip we treated the Staph aureus cultures with a bunch of different antibiotics, linezolid, clindamycin, ciprofloxacin, and vancomycin, and you can see that you can easily get these IC50 curves from such analysis. And also, if you take a look at this, even here it forms really nice biofilms, which is three-dimensional, has this three-dimensional structure, basically releases exopolymeric matrix, and also really viable cells. And we showed the same thing with Pseudomonas, and as you can see, it forms these really nice mushroom-shaped colonies, all encapsulated in a matrix. It is three-dimensional. And we combined, after that we also looked at polymicrobial biofilms, where we combined Candida with staph, because it is not one bug, one disease anymore. It is a combination of different bugs that can produce diseases. So you have these filaments, which are Candida albicans, and these colonies, which are Staph aureus, and together they coexist. And when we grew them in the chip and we tested against a bunch of antibiotics, so you have, let's say, a polymicrobial infection, you treat it with a bunch of antibiotics, you're not going to reduce the overall pathogen load. Or if you treat only with antifungals, like amphotericin or fluconazole, you're not reducing the pathogen load either. Because in this case, you're killing only the bacterial component, and in this case, you're killing only the fungal component. So you need a combination of both. Particularly, you need amphotericin and vancomycin together to kill everything. And with fluconazole, it is not effective because it is only fluconazole plus vancomycin. Fluconazole is not effective against biofilms. Okay, so this is another example of where you can use the chip to look for polymicrobial infections. So switching gears a bit. So we showed you this data. This data is all about drug discovery, where you're looking for new drugs. Okay, so I'm going to talk to you real quick about antimicrobial susceptibility test, and where we show that the same platform can be used for diagnosis. Okay, so like the previous speaker was talking about, in the antimicrobial susceptibility testing and microbial ID, typically when somebody checks into an emergency hospital, I could be wrong, so just bear with me. Somebody checks into a hospital, essentially you draw cultures, and then they are expanded, they are plated out, and then they are from the plates, which could be for selective growth. You essentially take out a colony and then test it against a bunch of different antibiotics, and then you record it. So the process basically takes about 2 to 3 days. Okay, and which is very crucial when somebody checks in with sepsis or one of these really nasty diseases. So this is how it looks. So it's a clinical microbiology lab, and they typically use systems like Vitek2, where from this you take a colony and then load it into the system, and you run the assay. Okay, this is taken from the county hospital in San Antonio. However, this may not be feasible in all cases. So this is one of the cartridges that they use. Okay, so this may not be feasible, especially if you're in a low resource settings. Okay, so what we did here is that we took the same idea where we had the microarray chip but flipped it. What we did here was we took the chip, okay, and on the chip we spotted the drug antibiotics at different concentrations. Okay, and now you have a slide with dried spots of antibiotics at different concentrations, and this can be stored for up to 2 weeks. Okay, and what you do is then, if somebody comes in with a sample, then you take the sample and spot right on top of each of those spots. Now, if the drugs are effective, it is going to interact with the bacteria, and it is going to kill the bacteria. And once you give, let's say, 24 hours, you can essentially read it with the scanner, just like I mentioned to you before. And for example, this is just an example of where you have no cells control, and you have different concentrations of the antibiotic in the bottom, and that essentially kills the cells. Okay, so we tested this against wild-type Staph aureus, which is in red, or against MRSA, which is in green, showing that the MRSA is more resistant to methicillin. So we have moved on from there, doing it on a glass slide, to paper, actually. So we used a paper-based platform for microbial growth, and the way we, instead of using a spotter, which may not be feasible in a low resource setting, is that we used just a printer to print wax on paper to create channels and spots which are hydrophobic, okay, so that you can confine liquid to certain locations, because all the rest of the area is all spotted with, is all covered with wax, okay. So the idea is, let's say so you want to have, you have a paper that is pre-printed with selective microbial growth media. Okay, so essentially what you're doing is a petri dish, but you're transferring the whole idea to do everything on paper. Okay, so I have a paper that is printed with media, and I could have different types of media in 1, 2, 3, or 4, as you can imagine. Okay, now I will put a spot of this here right in the middle and allow it to go to different locations. Okay, and you allow for about 24 hours or so. The things are going to grow at different spots or not grow depending upon what you have, and that will tell you what kind of organism you have, whether you have staph, or depending upon what organism you have, that will grow only in selective media. Okay, in parallel you can also have these spokes, and you have 16 drug loaded zones. For example, you could have different drugs loaded in different spots. Now you put your little sample here, it is going to go to these different spokes, and it takes only 1 minute to distribute. You allow it to, you wait for 4 hours or so, then the cells are going to either grow or not grow, and then you can read off directly, for example, with a iPhone app to see where the cells have grown or not grown. So this is really a low cost alternative to what is being done in the clinic. So we are still working on this, trying to make it better. That's where we stand. And the idea is to grow the optical readout, as I said. So you can see it's very faint, but yeah. So you can see that once we have an optical readout by staining, you have certain differences in colors that show up here. So obviously this idea needs a lot more optimization, but we are still working towards it. So this, just to sum up, essentially what we hope to achieve is, this is with currently existing techniques, which takes about 1 to 2 to 3 days. What we hope to achieve with our platform is microbial susceptibility testing platform is within about 8 to 16 hours, so that when the shift changes, we should be able to provide them with the results, and also a much cheaper alternative. So these are people I worked in the lab, and particularly for this project, I would like to thank my collaborator, Jose Lopez-Rebord, and Chris Frey, who are microbiologists. And as I said, almost exclusively this work was done by one of my graduate students who finishes PhD now, Anand Ramasubramanian. So, and I would like to thank NIH and DOD for grant support. And I will take any questions. Thank you.